This is Mr. Holsey with H Squared, Sufficiently Understood Science, and today we're going to be talking about Punnett squares. So let's talk about human chromosomes first, because chromosomes are your genetic makeup. They make you who you are. And so we're going to take a look at chromosomes for just a moment. They're these little X-shaped kind of things. And so human cells have 23 pairs of chromosomes. We get one from mom, one from dad. So this word pairs, meaning times 2. So you get 23 of your chromosomes from mom, 23 from dad. That comes together and makes you with a total of 46 chromosomes. Now, for the most part, all these chromosomes are the same regardless of gender, male or female, except for the 23rd pair, which determines your gender. You either inherit two X-shaped chromosomes, which makes you female, or an X-shaped chromosome and a Y-shaped chromosome, which makes you male. So what if you wanted to be able to predict the outcome of two breeding partners? Well, it's actually really simple, and you can use a very basic tool developed in 1905 by Reginald Punnett in order to basically understand what the percentage chance of an offspring having a certain phenotype is going to be. So this is devised, like I said, by Reginald Punnett in 1905, and it's a method for finding the probabilities of possible genotypes of offspring of a cross or breeding experiment. This diagram method is known simply as the Punnett Square. So the alleles, which you can see right here, these are the alternating forms of the same gene. So here we have a capital letter, which represents a dominant gene. And over here we have a lowercase letter representing a recessive. Now you're not always going to have it in this pairing, but just giving you an idea. So dominant genes are always going to be expressed.
Go ahead.
While a recessive trait is not always going to be expressed. It is only expressed if there's no dominant trait. So inside these boxes, we have 4 potential offspring. 1, 2, 3, 4. And each square represents a 25% chance of what that offspring will look like. So let's talk about the difference between heterozygous and homozygous. So a heterozygous a heterozygous parent has 2 different alleles for a single trait. Let's say eye color. It's the traditional one that a lot of people are very easily understand. Brown eyes versus blue eyes. Brown eye color is dominant over blue eyes. Blue eyes is a recessive trait. So that means the parent has one dominant gene and one recessive gene. Homozygous means that has the same 2 alleles for a single trait. So once again, going back to the eyes, if we have 2 capital letters, that parent only has genes for brown eyes. If they have 2 recessive alleles, they only have genes for blue eyes. And so this is actually how it makes it very easy to understand what's going to happen. So to show you really quick what these pairings look like. So if we were looking at heterozygous, we're looking for 2 different alleles for the same trait. And so both of these are heterozygous because they have 2 different alleles, a dominant and a recessive. Whereas on the other side, both of these are homozygous. The two capital letters would be homozygous dominant, and the two lowercase letters, homozygous recessive. So remember that genotype determines the phenotype. Dominant traits express over recessive traits every time. And so phenotype is the outward appearance of an organism. So for example, if my genotype was two dominant traits, my phenotype would be expressed dominant. Okay? If I have one dominant, one recessive, also known as heterozygous, then my expression, because I have that dominant in there, my phenotype will express as dominant. The only time you're going to see recessive traits show up in a phenotype is in a homozygous recessive pairing, where there's no dominant allele to overcome the recessive allele. And that's not to say that recessive alleles are bad. Basically, it's just saying that they show up less than dominant alleles. And so here that I have two recessives, I'll actually receive a recessive trait in my phenotype. So using a Punnett square is extremely easy. And one thing I like to do is kind of figure out like how I got my blue eyes, and I'll do that one for the last one. And so very basic 1, 2, 3 set of rules. So each parent's combination of alleles must be placed on the same side, on the top and left side of the square. And I'll show you how to do that in just a minute. Each box represents a 25% chance the offspring will receive that genotype. And simple enough, move one letter at a time. Don't rush through things. So our first pairing we're going to do is going to be a homozygous dominant, and we're going with blue eyes, crossed with a, let's just go ahead and do a heterozygous. So a homozygous dominant, I'm going to use the letter A for mine, is going to have two capital letters. A heterozygous will have one dominant, one recessive. So rule number 1, each parent's combination of alleles must be placed on the same side. It doesn't matter what parent I put where, as long as I keep them the same. So this is going to be parent 1, this is going to be parent 2. I'm going to go ahead and label those as 1 and 2. So I'm going to put parent 1, capital A, capital A. So there's parent 1, my homozygous dominant. Heterozygous, parent 2, capital A, lowercase a. So this is a very simple process. All it's doing is bringing it down and over. So I'm going to bring this over. So I have A, A. I'm going to use a different color to show that. Bring this over, bring this one down, and I end up with dominant, recessive. And do the same thing, over, down, dominant, dominant, over, down, dominant, recessive. So I'm going to get rid of these arrows really quick and then make it a little more clean. So it's a very simple process, just bringing things over and down. And so if I look at this, each one of these is a 25% chance that the pairings will have this type of genotype. So the chances of a dominant phenotype being expressed in this example is actually 100%. Because each one of these boxes, well, each one of these combinations has at least one dominant trait in them. So there's a 0% chance that these offspring will have a recessive trait showing the phenotype, but not that they can't pass that on. Okay? Let's say of this pairing, this one right here is the one that is born. Okay? So we're going to skip ahead some generations. So we had a heterozygous born from this pairing. They show that dominant trait, and later in life, they are going to have children with someone who doesn't show that trait. In fact, let's say we have a homozygous recessive, which is characterized by two lower cases because they have a phenotype that shows a recessive trait. So I'm going to put parent 1 over here, a, little a. Parent 2 from our original generation, capital A, little a. And I'm just going to do the same thing. I'm going to go ahead and make the arrows to show you. Where these are going to go. All right. I bring them down, I bring them over. So I'm going to bring over my A here, my lowercase a. I'm bringing down my capital A. So we got our heterozygous. I'm going to bring it over right here. Got a homozygous recessive. Bring it over, bring it down. Oops. Bring it over, bring it down. And so now, remembering that this is a 25% chance for each one, okay, 25% chance, I can determine how many of these offspring have a chance of being dominant expression in their phenotype or recessive expression in their phenotype. So I have a dominant trait here, and I have a dominant trait in this one. And so we're right down the middle with this one. So dominant is a 50% chance, while recessive is a 50% chance. Because neither of these two, let me get a different color really quick. Neither of these two have any dominant traits. That's the only way a recessive trait can show in a phenotype is if there's no dominant to overshadow it. All right, we're going to do one more example really quick. And this is my favorite because this expresses me, how I got my eyes. So I have blue eyes. And both my parents have brown eyes. So the question is, how did I get that? Because in our first example, okay, we had a homozygous dominant with a heterozygous, and there was no chance for a recessive phenotype coming out of their offspring. Our second example was a homozygous recessive with a heterozygous, but that would mean that one of my parents had to be blue-eyed, which they were not. And so how can I get that? With them both showing dominant traits. And so in this pairing, we're going to have a heterozygous cross with a heterozygous. So we're going to place this on our Punnett square, parent 1, parent 2, and we're going to bring it over, bring it down, bring it over, bring it down, bring it over, bring it down, bring it over, bring it down. All right. And so we're going to go back to our percentages here. And so there was, looking at the dominant traits, 1, 2, 3 of these boxes, which once again, 25%, or 1 in 4. 3 of those boxes have a dominant, at least one dominant trait. And so dominant expressed phenotype is going to go up to 75%. 75% chance that I should have had brown eyes. But right here, we can see we have a homozygous recessive. And so there's a 25% chance of getting blue eyes. And this pen is not wanting to work right now. So 25% chance of having blue eyes. And that's where I got my eyes. Now, fun fact, my wife also has blue eyes. And so it wasn't even hardly a guess as to what our children's eyes would look like. Because now, since blue eyes only show with recessive traits, let's say we had two homozygous recessives pairings. Oh, there it goes. There's no percent chance for a dominant expression and 100% chance for recessive expression. This has been used by farmers and scientists for a really long time because getting a dominant trait out of a population versus getting a recessive trait out of a population is two very different stories. Getting rid of recessive genes take a really long time because they hide, whereas dominant trait genes are very easy to selectively breed out of a population, which will be the topic of a later video that we do. So once again, this has been Mr. Hulsey with H Squared. It's officially understood science. Remember to like, share, and subscribe, and check us out for more video tutorials. We're going to start a new series working with 7th grade tutorials. So looking forward to seeing you next time.